I've always admired Viahart and how her fantastically fast fingers fly frenzied from drawing to drawing, so for this project I'd like to credit her for inspiration and thank her for making my gravitational biology video a little bit more interesting. I hope you can give her channel a watch. Phase 1, Signal Perception. Phase 2, Signal Transductance. Phase 3, Gravitropism. 3 phases defined in plants' perception of gravity. This video will aim to provide you a little greater understanding of each one. I've got 3 to 4 minutes and we'll be going fast, so before we get into how a plant orients themselves with gravity, I want to get a few definitions out of the way. Ready, go. Biochemical. This is in reference to chemicals used by biological systems. Sedimentation or sedimenting usually refers to rock formations, but in our purposes it means an object settles on the surface or one another. Transduction, a process of converting physical signals into chemical reactions, specifically so that biology can know what's going on with their environment. Tissues, a collection of specialized cells. Anything with the prefix gravi means that is related to gravity. An example would be graviorientation, which relates to how organisms' orientation is related to gravity. Quick prefix practice. Gravitropism. Tropism means to grow in a direction, hence gravitropism means growing towards or away from the gravity vector or the direction of gravity. Presentation time. The time stimulus needs to be applied before a reaction takes place. Centrifugal force, an apparent force acting outwards of a spinning object. Centrifuge, a machine to produce the centrifugal force. Okay, with definitions out of the way, it's time to talk about the one Homo sapien no biologist has ever heard of, Charles Darwin. One of the earliest to write about the phenomena of how plant root caps' integrity relates to gravity. When the root caps are damaged, the root appears to grow agravitropically or independent from gravity until the root caps have reformed. This was an early step in understanding the gravity sensing of a plant, indicating that orientation of the root growth likely utilizes specialized tissues. A zoomed-in perception of the root tip growth shows many layers, the bottom of which has been labeled the columella. Later research has found that these cells are particularly effective when disarming orientation. Our current understanding of how plants orient towards gravity is named the starch statolith hypothesis. In normal gravity, it works similar to grapes in a cup of water. In our case, the grapes are called statoliths, and they are the only objects free to move about the whole cell. As a whole, the cells are called statocytes. Each of these statoliths rests on a cellular structure called endoplasmic reticulum, or ER for short. There are 2 types of endoplasmic reticulum in a statocyte, one that rests on the bottom like a little bed, and the other lining the cellular walls. The statoliths usually sediment towards the ER bed when the cell is perpendicular to the gravity vector. And this is the nucleus. That usually stays here, but we draw it because it's so big and gets in the way. If you take a cup of water containing grapes and tilt it, the grapes sediment similar to how statoliths fall and interact with the endoplasmic reticulum. According to the hypothesis, phase 1 signal perception begins here. Calcium ions are likely the beginning of the next phase, termed signal transduction. In this phase, the organism translates the signal from physical stimulus to biochemical reactions, allowing the plant's systems to respond. Calcium ions are thought to have been used because they have been found contained in the ER, and manual addition of calcium ion leads to the root bending towards the application site. After stimulating the ER, calcium ions are likely released, beginning a biochemical chain reaction. A slew of secondary chemicals flood in to carry the message across the plant, regulate new surplus of calcium ions, and aid in the phase 3, gravitropism. In this case, gravitropism is referring to the act itself of growing in orientation to gravity. Due to the accumulation of ions, growth is inhibited on the bending side. Growth continues on the opposing side and the root bends. The end result is returning the root orientation towards the pull of gravity. To recap. Step 1. Statocytes and statoliths are at rest. Step 2. Shaking, tilting, manipulation causes the statoliths to agitate the ER. Step 3. Calcium ions are released and is likely the beginning of a biochemical chain reaction. Step 4. Growth is inhibited on the calcium ion saturated side, and the secondary messengers aid in growth on the opposing side. Step 5. The plant system begins to regulate itself. Gravitropism slows and eventually ends. Those are the basic steps found in the starch statolith hypothesis. Centuries of research have been conducted to determine so many of these processes, and much of our current research has been omitted in favor of time restraints, but I'd like to point out a few notable exceptions. Sir Thomas Andrew Knight in the early 1800s published his findings on the plant root gravitropism. He found that placing a plant in a centrifuge causes the plant's roots to grow towards the sum of the centrifugal force and gravity, meaning that plants likely grow towards the acceleration, not gravity in and of itself. Another is in the mid-1980s when research aboard the space shuttle showed that the presentation time of gravitropism would increase twofold when in microgravity, likely due to the statoliths not having sedimented towards the bottom. If you'd like to learn more about gravity sensing and how plants, fungi, and protists react to gravity, please read Gravitational Biology 1, Gravity Sensing and Gravity Orientation in Microorganisms and Plants by Marcus Braun, Mayank Bomar, Donnet Peter Hayter, Ruth Hemmersbach, and Klaus Palm. This has been a brief summary of Chapter 6, Gravitropism in Higher Plants, Cellular Aspects. I would like to thank the researchers I used in this video for sharing their works and their findings.